The next speaker is Nikita Vladimirov, who is a staff scientist at the University of Zurich, who'll be telling us about Benchtop mesoSPIM, an open source light sheet system for imaging clear tissues. So we're now going down a step in scales from whole human imaging to tissues imaging. Nikita, please go ahead.
Hi, thank you very much for this nice opportunity to spread the word about open source systems. So my talk will be about open source light sheet microscope, and this is the example of the data that we get. So it's much smaller samples and also high resolution. This is a mouse brain cleared with VDisco protocol and imaged along with its spinal cord. And the system that I'm developing is called mesoSPIM project, and particularly the latest version called Benchtop for its smaller size. And this is the CAD model of the system. And just to bring everyone to on the same page, in my particular field, there are two techniques that really revolutionized the microscopy in the last 20 years. It's light sheet microscopy and tissue clearing. And so these two techniques, they kind of push each other to new challenges and boundaries. So the light sheet microscopy is different from other microscopy modalities in the sense that it eliminates only one plane of a sample at a time. So there is no out of focus light, there is no bleaching of other planes like in a confocal. So it is very fast because it's a two-dimensional image captured at a time. And it gives, so as you scan your sample plane by plane, which you can do very fast, you acquire three-dimensional image really fast. So it gives high contrast, high speed, high optical sectioning, and low photo damage. And the other technique is tissue clearing, which allows you to chemically remove lipids from biological tissues and essentially make them transparent like glass. So the brains, for example, a mouse brain on the top image, it's just a fixed mouse brain, and after removal of lipids, it becomes transparent, but it retains the fluorescence, the fluorescent proteins and labels that are labeling some proteins of interest or other biological structures. And so these two techniques, they're really made for each other. So they together they really revolutionize our field. And another development of the light sheet technique, which is really useful, is called axially swept light sheet microscopy, or ASLM. So in more traditional light sheet microscope, you have this Gaussian beam of laser light focused, and the most useful part of this beam is here in the center near the focus point. That's where your sample, your Z resolution is the highest. And so if you apply it to a big sample like a mouse brain, then you have a very nice resolution in the middle, but very poor on the periphery. However, if you refocus this beam, so you move the focus from left to right, and you use a special camera mode with a rolling shutter, so you acquire only one band at a time, you can have a nice Z resolution across the centimeter scale of the sample. And the mesoSPIM project uses this technique, and it was started by Fabian Voigt in Friedrich Helmchen lab in 2015 as a little side project, and it grew up into more global global open source project. So it was open source from the very beginning, and the version 5, the big microscope, was published in 2019. And this year, so I joined the project relatively late in 2021. And this year, we published the newer version of mesoSPIM, which is called Benchtop. It's much smaller, has high resolution and other advantages. And so all the sources are free on the internet, on our websites. And it's open source. It's licensed as a GPL project, both software and hardware. And this is the distribution of the systems around the world. The ones we know of course, people who build it are not obliged to report us back. And there is a growing number of application papers, especially in neuroscience, but also in other biomedical fields where people are interested in anatomy, especially. So I will probably quickly go through specs. So it's a not high resolution technique. It's about 1.5 micron by 4 micron axial lateral versus axial resolution. It's very fast. It can scan the entire mouse brain in about 15 minutes. It has a large field of view. It works all with pretty much any popular clearing method. And its cost is relatively low compared to commercial systems. So the version 5 is 160,000 and Benchtop is below $100,000. And the Benchtop has several key advantages from the previous version. It uses newer generation cameras with larger sensors, so larger field of view, larger magnification, smaller footprint, a lower cost, and it is easier to build. So we really aim at non-experts, non-engineers that should be able to build it. And it can be mobile. For example, the first Benchtop that we built is now traveling in Bordeaux in Neuroscience School. So you can just, yeah, this is the summary of the differences. I will skip through it. One of the things that was developed for Benchtop is a new objective testing technique so that we know precisely what objectives work best for our light sheet imaging. And it is a quite simple technique. It images gratings, different defocus, and then we analyze these images, these three-dimensional stacks, and we calculate the contrast maps of this objective, and each objective has its own distinct contrast map. And then by quantification, we know what to expect from this objective in terms of, for example, field flatness, overall contrast. So this allows us to quickly select for objectives that work best in our application. And this is the sample that I showed in the beginning. So this is a cleared mouse brain. We put it into the microscope and acquire the images by tiles. Then we stitch tiles. And these are different snapshots, max projections, which show single cell, even axial resolution of the system. And this is magnification 5x. So it's not a really high magnification system. And we tinker with a lot of different samples. So more and more people want to try their samples. So we 3D print and share the designs of various sample holders. And these are some examples of what we can image. So on the left, this is the same mouse brain I showed before, but at magnification 20x. So you see axons very well for pyramidal cells and basal dendrites. And on the other range, at lower magnification, less than 1.9, you can image the entire mouse, cleared mouse, young mouse. And there are more high throughput techniques. So for example, we developed this tower, which allows imaging of many tadpoles or other relatively small samples and quantify the development abnormalities of tadpoles depending on the treatment. And some unexpected applications from physics recently popped up. We have users from physics department who study dark matter particles and they irradiate crystals of calcium fluoride, and the crystals work as a passive particle detector, and when there is a high energy particle, it leaves a fluorescent trace in the crystal, which we can image And this is other microscopes cannot achieve this depth of imaging and this volume in a reasonable time. So we also support those people who already have older versions of mesoSPIM microscope, so they can upgrade to the newest objectives and newest cameras. Our software is also all open source. We write it ourselves in Python, so it's also available for free online, so people can just upgrade their system and change config file to work with new hardware. And in summary, the mesoSPIM is open source, do-it-yourself facility grade light sheet microscope. It works best for intermediate resolution and fast imaging of large cleared samples. If you decide to build, the lead time is about 3 months, and it's mostly waiting for parts from vendors, and construction takes several days. And it is cheaper, easier to build, and gives high resolution over a larger field of view than previous generation. And in the near term, my near-term goal is to achieve even higher resolution, larger sensors, and high throughput, especially inspired by recent publication from Allen Institute by Adam Glaser, the mesoSPIM, which really pushes the field in that direction. More automation and less user training through smarter software that we develop in-house, and definitely more documentation and outreach to really bring new users and new builders. So these are two separate crowds of people, typically builders and users, but they are both essential for growth of the project. And my key takeaway probably is that mesoSPIM is not a fixed microscope which you can just buy. It's a different mindset. It's a framework. You can modify it, you can upgrade it, you can disassemble it if you want to build something else. So it's really you own the system, and it's very modular and upgradable, and it both software and hardware. So it is a framework rather than just a fixed setup. And the project was made possible by really many people. Of course, Friedrich Henken and Fabian Voigt as the founders, but also by other collaborators, both local and global power users, builders, contributors, and supporters. So it really takes a village to raise a child, but also to build such an open source microscope. This is the QR code for the Benchtop publication, currently in review, but it's available via archive, and just some links and resources to our websites. And if you decide you want to get started, but you're not ready to risk $100,000 yet, you can just clear your samples, come to Zurich, get training with us. It takes about 2 hours, and image them yourself. Just contact me. Thank you very much for your attention, and if we have time, I'm open to questions.